Hello, we are going to start in a few minutes. Okay. So, hello and welcome everyone to today's live class session of week 10 on the course Experimental Biochemistry, which is being taught by Professor Shagata Dasgupta and Professor Shomodaya of IIT Kharagpur. I am Srijan Sen, and today I will be taking your live session on the courses which were taught to you in week 10 and also discussing with you a few questions which will also help you in solving the assignments and taking up the exam which is coming up. Okay, so let us begin. I am sharing my slides with you people. So I guess my screen is visible to you all. So let us begin. So basically in today's lecture, we will be studying or hearing about recombinant DNA technology. What are the applications of it? What is how we do recombinant DNA technology? What is recombinant DNA? We will be knowing about it and mostly the techniques involved in it, what you have to do, how we do things, stuffs like that. So we will be studying about that. Okay. So we all know about this, that is your DNA, that is your ribonucleic acid, deoxyribonucleic acid. So it is basically a very large molecule which is made up of your smaller units which we call, or the basic units which we call as the nucleotides. So these are so this DNA is made up of your nucleotides. Then these nucleotides have three parts which we all know is a ribose sugar, then we have a phosphate molecule and nitrogenous bases. So the nitrogenous base is the part of the nucleotide which carries the genetic information. So this is important because the nitrogenous base is actually the one which carries your genetic information. So this is responsible for storing your genetic information and carrying the information. Okay, so the bases which are found in DNA are basically four. Your adenine, then cytosine, guanine and thymine. So among these we call it as adenosine triphosphate, cytosine triphosphate, guanine triphosphate and thymine triphosphate. So these four of the bases are known to us, and these are the general four bases which are present in DNA. Whereas in RNA we do not have the thymine, we have is what we call as the uracil, right? So this is the base pairing which occurs. So we have heard in the previous class about the base pairing and all, like if your adenine forms double bond with your thymine, then cytosine forms triple bond with your guanine. So we have heard about it. And then we have the sugar, and then the backbone is made up of your sugar and phosphate. So this is what the backbone is made up of, and then we have the different hydrogen bonds which are involved in your base pairing effect, and these are basically your nitrogenous bases. So we know this, and then we have adenine, thymine, cytosine and guanine. So what is a gene? So we all know that it is a stretch of DNA that codes for a type of a protein that has a function in the organism. So it is basically the unit of heredity in any living organism. So all the living organisms depends on this genetic material, or what we call as the gene. Now these genes are involved in carrying or storing of informations that are important in building and maintaining your say body cells or any cells present in any organism, and these are the informations which are passed on from generation to generation, like the passing of the genetic traits to the offspring, from the father or the mother to the offsprings. Okay, so basically your gene is it's a long stretch of your DNA which performs the different function of in an organism. That is, the function involves the building and maintaining the cells in an organism, and then passing the genetic information to the next generations or to the offsprings. So what are the gene components present? So these genes contains, so you have different things like your promoter, where your RNA polymerase binds and the protein starts expressing and all. So you have the promoter, then you have the exon, then you have introns. So these are the different things present. So these exons are responsible. So these exons are basically known as the coding regions, which codes for the different proteins, and these are the parts of your gene which are involved in carrying the genetic information and then forming of the proteins which will actually help in the functioning of the body, or which are responsible for different body functions. So these exons are very important, and these are the set of coding regions which codes for the proteins, whereas the introns on the other hand are the non-coding regions, and these are the non-essential part, and therefore what they do, they splice out of the gene product, and then the exons attach, and then via that we get our protein in our hand. Okay, so these introns are the non-coding regions, removed sequence, and are therefore labeled split gene splicing. So these introns move outside, the exons join together, and then we get our protein. Okay. So what is a genome? So the genetic complement of an organism, including all of its genes as represented in its DNA, is termed as the genome. So it is the genetic complement of an organism that is present, that is, it includes all of the genes as we sequence it in the DNA. So this is known as the genome. Next, coming to the gene expression. So what is gene expression? So what happens over here is that it is generally the process by which you can synthesize a functional gene product, say proteins or something else you want to synthesize from a particular gene. So you are actually expressing your gene. So you are allowing the gene to showcase itself. So expression is showcasing itself. So you are allowing the gene to showcase itself. So what it can do, what it wants to do. So that is synthesizing, say, a gene product. That can be even your protein or something else from a particular gene. Now the process of this gene expression is used by your eukaryotes, then your prokaryotes, then different viruses. Why they do that? To generate the macromolecular machinery of life. So gene expression is done by all of these organisms to perform certain functions, that is by generating the macromolecular machinery of your life. Now what we see here is the DNA, then we go for mRNA transcription. After that we get the mature mRNA, and after that there is translation, and these are your TR mRNAs carrying your different codons coding for your particular amino acids, and thereby we get our protein of interest. So the different steps of your gene expression includes your transcription, which we all know, that is an mRNA synthesis. After that there is different types of post-transcriptional processes which are taking place, that is your RNA splicing and all. After that there is translation, that is synthesis of our proteins or the synthesis of the gene of our interest, and then the post-translational modifications of our proteins, which includes say phosphorylation, glycosylation, ubiquitinylation, etc., depending on what the proteins want to post-translationally modify themselves to. So here what we see is, so what we see here is, this is a DNA, double-stranded DNA, and after that we are synthesizing the mRNA inside the nucleus. After that what happens, the mRNA moves out of the nucleus into the cytoplasm via the different pores present in the nucleus, that is the nuclear pore, and then it goes into the ribosome where the synthesis of the your polypeptide or your proteins takes place. So the amino acids join via the peptide bonds, and then the polypeptide is formed. After that it refolds itself, so it can refold inside the ribosome also, or maybe outside it, and in the meantime it can undergo different post-translational modifications as well. So post-translational modifications occurs after the polypeptide has been formed. So these are the different steps which are involved in the expression of your gene. Now the next thing is that what are genetic changes? So any alteration in the segment of the DNA which is affecting a gene's behavior or the functioning of an organism, the different kinds of diseases which are observed in an organism can be termed as the genetic changes. Probably you do not see that in the father or the mother, but you see that in the offspring. So that is kind of a alteration which has occurred in the sequence of the DNA of that particular organism, and this, what it does, it you can see different say abnormalities in an organism, and also sometimes these abnormalities leads to different kinds of diseases. Like we have seen a lot of birth diseases, then diseases related to growth, and hormonal disease which affects the hormones and all. So these are the different diseases which can occur due to your genetic changes and all. So these genetic changes can be of two types. One is where the genetic change is very small, and there is a genetic drift which has occurred, and it's a very small kind of a genetic change leading to a particular mutation in all, such that you say, I mean, it's not for practice, but for example you're placing your hand inside the UV without your gloves and all, so then there can be a mutation in your skin cells and all, and that can actually cause cancer. So that's a small genetic change which is occurring. And the next one which we observe is the large genetic change which is causing the genetic shift. So your small genetic changes, that is a mutation, is causing a genetic drift, whereas your large genetic change, that is a recombination, is causing a genetic shift. So these are the two types of genetic changes which occur, and each of these factors are involved in affecting an organism's function and all. So even the different types of diseases can be critical depending on the type of genetic changes which have occurred in that particular organism. Now what is mutation? So the one first example that comes into our mind is that hemoglobin thing. So the hemoglobin DNA, and where the glutamic acid is converted to valine, and then we see the sickle cell uh, condition, okay, of these RBCs. So what happens over here? There is only a single mutation which is occurring, that is your thymine is getting converted to adenine, and in the same way the mRNA sequence changes and there is a change of codon, uh, the triplet codon, and then you will observe a change in the protein. So any changes in the DNA sequence of a cell's genome caused by, you say, radiation or transposons, some mutagenic chemicals, or the different types of microorganisms, viruses, etc. So there are different factors by which these kind of mutations can occur. So ultimately these mutations can be even smaller, like we see in the this is only a single nucleotide which has been mutated to something else, and then we are seeing a very diseased condition, from a normal hemoglobin to a sickle cell hemoglobin condition, or sickle cell condition, that is a diseased condition. Whereas what is your recombination? So recombination is the exchange of corresponding DNA segments between adjacent chromosomes during the special type of cell division that results in the production of your new genetic makeup. So in the previous case what we saw, it is just a single nucleotide which is being changed to a different type of a nucleotide, okay. But here what we see, we are changing the entire DNA segment, okay. So this is a large genetic, like we said here, the large genetic change which is causing a genetic shift. So this is what we are trying to convey. The message is that recombination is your exchange or just removal of a part of the DNA segment and exchanging with a different part, and it should be present between that part where we want to, where the expression of the protein will take place. So between the adjacent chromosomes during the special type of cell division that result, that is going to form or produce a new genetic makeup or a new genetic gene product. So that is actually recombination. So it's a large, it is changing the large DNA sequence, whereas a mutation is for a very small kind of nucleotide or a very small gene change. So what is this recombinant DNA? So the DNA molecules which are formed by laboratory methods of genetic recombination, such as you say molecular cloning, to bring together the genetic materials from multiple sources, thereby creating sequences that would otherwise not be formed in the genome. Say you want to add a particular special type of an amino acid, or you want to mutate an amino acid and check the functioning of that particular protein, that you are mutating a few of the amino acids and then changing, or you are changing the entire of the gene sequence, and then you do molecular cloning and all, and you bring the genetic materials together, you join them, and then you check how your protein is functioning after all those mutations and the necessary changes which you have applied, and how your protein is reacting to it. So those are the different ways. So the recombinant DNA is that thing. So recombinant DNA is possible because DNA molecules from all organisms share the same chemical structure. They differ only in the nucleotide sequence within that identical overall structure. So this point is important. So why is recombinant DNA or is possible? Because first of all is that the DNA molecules have the same chemical structure and they differ only in the nucleotide sequence. Okay, so that is why it is possible to make recombinant DNA. Now, this recombinant DNA is the general name for a piece of DNA that has been created by the combination of at least two strands. Okay, and these recombinant DNA molecules are often called as the chimeric DNA because they can be made up of two different kinds of species materials like the mythical chimera, and it uses the different types of techniques for making up so it is useful for making up the materials from the two different species, giving it and say a magical type of a sequence and all, or the chimeric type of a sequence. So you're changing it. Okay, so it is the general name for a piece of DNA which you create by the combination of at least two DNA strands. And what can we do using this recombinant DNA technology is that we can isolate and clone single copy of a gene or a DNA segment into indefinite number of copies, all of which are identical. So clones means the same number of copies of the same identical copies of the same DNA segment. So which we do by the PCR. Now, these new combinations of genetic material or recombinant DNA are then introduced into the host cells, where they are allowed to grow and multiply. And this entire technique is termed as your recombinant DNA technology. Now, in this we have the different restriction enzymes which are responsible for cutting at the different sites. So what these do, these restriction enzymes are basically your bacterial enzymes that cut long strands of nucleotides into the smaller segments. So what they do, then they form a linear kind of a vector and all, into which you are going to insert your DNA, and then you join them, and then things go about in the same manner as. But what you are doing at first, you are just cutting them in a particular site and removing the DNA which is present over there and exchanging with it with a different kind of a DNA. Okay. Now, so what these restriction enzymes do is that they recognize specific sequences of nucleotides and cut the DNA at a specific site within the sequence. Now, in one chain, the sequence runs left to right, and on the complementary chain, the sequence runs right to left. And this is not much important. And your so this is a property of your restriction enzymes, and these restriction enzymes are recognize the palindromic sequences. Like in eco R1 you have GATTC, and in the reverse way you also have GATTC. So it cuts just after the G in both the sides, and then you get a linear kind of a vector, and then after you join you get it. You can also see from this image also. So these single chain tails of DNA called the sticky ends are created on each of the DNA segments by the action of the restriction enzyme. So if it is say it is cutting at a particular end, then you will see what you observe is that you have a part which is left over here, and you have a part which is left over here. So these parts which are left over and are left uncut are known as the sticky ends, whereas if there is a symmetric cut, then we call it rough ends, or like that. So after the sticky ends are formed, which will actually help in the joining of your DNA segment when you also cut them with the restriction sites and all. Okay, so these sticky ends readily bind to the complementary chains of your DNA. So the pieces of DNA that have been cut with the same restriction enzyme can bind together to form a new sequence of nucleotides. So when you are designing your DNA, you should have the say your restriction sites present over there, and then you cut these DNA segments also with these particular restriction enzymes and all, and then you can join them using some kind of enzyme called the ligase, DNA ligase. Okay, so these restriction enzymes can be used to isolate or cut out a specific gene. Now there are different kinds of vectors, one of which includes your cloning vectors. So this is a DNA carrier used to clone a gene and transfer it from one organism to the other. Now, once you have isolated the particular gene, uh, then you can place it inside the cloning vector and then introduce inside the host cell of or an organism. Okay, now bacteria contains circular vectors, or you can call that as a plasmid. So the circular vector is called as a plasmid, which is present in many bacteria. Now what happens, these plasmid vectors, then you have some DNA fragments which are to be cloned. Now what you do, you use the different restriction enzymes to cut these plasmid vectors at different sites. So these restriction enzymes only represent specifically particular sites, which are your palindromic sequences. Now they cut only at those particular sites. It's not like random cut. EQR1 cuts at a random position, then your HIN3 cuts at a random position. This is not the story. The story is that the restriction enzymes cut at specific sites, and then at that particular site your DNA will also get cut, and then it will help you in joining. So now as of now, the plasmid vector, you cut the plasmid vector with particular restriction enzymes and all, and the same treatment is done with your DNA fragments which are to be cloned, and then you insert the DNA fragments into your plasmid vector. It takes up and then you transform them into your E. coli cells containing any kind of, you know what your vector is resistant to, and then only those bacterial cells which have taken up your plasmid with those sequences will only be uh, resistant to that kind of antibiotic and will grow in your agar plate. So plasmid is a ring of DNA found in bacteria. In addition to its main chromosome, it can be used to transport genes from one organism to the other. And this is your electron micrograph of a plasmid. You can see it's a circular form in a double-stranded DNA form. And so this is all. Now, how do we use a cloning vector? So a plasmid is removed from primarily from the bacterial cell. Then you use a restriction enzyme to cut it, and then you introduce a donor gene. So what you do at first is that you cut the plasmid in the in specific sites, and then you do the same treatment with your DNA fragment as well. And this plasmid contains sticky ends, and now so your DNA fragments are also going to contain your sticky ends. So now these sticky ends come close to each other, and then they form your recombinant DNA. And obviously there can be different kind of mismatches, like your two vectors can come close to each other and join, two DNA fragments can come close to each other and join. So but these kind of things also are the side reactions which occur in the same time, but only your recombinant DNA is going to grow inside your plasmid, because is going to grow on your agar plates, because they contain some kind of resistance. Sorry, your yes, some kind of resistance to the different antibiotics, and only those cells which are taken up by your, or only those plasmids, recombinant DNA plasmids, which are taken up by the bacteria can only grow, because they can your express the different antibiotic resistance things and all. And after that, what happens, the plasmid is returned to the bacterial cell, where it replicates as the bacterial cell divides. Now this clones the donor gene, and the bacteria containing these clones of the donor gene can be used to infect other organisms and transfer the gene to them. So here you see a plasmid vector is there, and the DNA fragment you enzymatically insert it, and then you mix the E. coli cells with the plasmid in the presence of CaCl2. So this adds to the competency of the cells, and the cells becomes competent in nature and can withstand high amount of stress, temperature and pH. Now the culture on the new agar plates containing, say, ampicillin as of now. So you have the bacterial chromosome and the plasmid which is transformed. E. coli cell survives. The cells that do not take up the plasmid die on ampicillin plates. So only those cells which take up the plasmid undergoes your plasmid independent plasmid replication, and then there is plasmid multiplication, and after the multiplication is done, the colony of cells is what we observe on your agar plate. So these colony of cells each contains your copies of the same recombinant plasmid. Now in some cases it happens that the plasmids are used to clone a specific gene so that the bacteria will produce a specific kind of a protein. Like we have heard of insulin, which is a protein that controls your sugar metabolism. So in the same way the treatment is done, and then it is cut at different sites by particular restriction enzyme. This plasmid is cut and then you insert the human cDNA into it via the different kinds of linkers. Now here you get, via your enzymatic reaction, you get your recombinant plasmid, and then what you do is transfection of this recombinant plasmid into the bacteria, where and then you get your insulin gene inside your bacteria, and then you can express it and get the insulin protein. Now how do we obtain this recombinant DNA? So these are the different steps. So in the first step, what we do, the DNA fragment containing the gene sequence to be cloned, which we also call as the insert, is firstly isolated. So firstly you isolate these DNA fragment, after which you cut the DNA and then you join the DNA. Now after this, what we would do in the fourth step is we insert these DNA fragments into the DNA fragments into the host cells using a vector that carries, so the vector is one that carries a DNA molecule, and then the rDNA molecules are generated when the vector self-replicates inside the host cell. Now in the sixth step, what happens, the transfer of the rDNA molecules is done into your appropriate host cells where you want to transform it, and after that the selection of these host cells carrying the recombinant DNA is used as a marker, and after this the replication of the cells carrying your recombinant DNA molecules can be used to get a genetically identical cell or a clone. So in this way we obtain the recombinant DNA. Now we isolate it. So the first step in making the recombinant DNA is to isolate the donor and the vector DNA. So the procedure which we use over here for obtaining the vector DNA, it depends on the nature of the vector and all. Now the bacterial plasmids are the commonly used vectors, and these plasmids must be purified from the bacterial genomics DNA. So this is the process of your isolation. Now after you have isolated it, you need to undergo ultracentrifugation. So previously we were taught about ultracentrifugation techniques and the different centrifugation techniques, at what speed and for what time you need to centrifuge it such that you get the different cell organelles and all. So depending on that, the protocol or the principle of this ultracentrifuge is basically the same. So a protocol for extracting this plasmid DNA can be achieved by your ultracentrifugation. Now the plasmid DNA forms a distinct band. Okay, after you have performed ultracentrifugation in this density gradient using a cesium chloride which contains your ethidium bromide. Now why we use ethidium bromide? Because it intercalates inside the double-stranded DNA, which you can observe under the UV light. Now the plasmid band is collected by punching a hole in the plastic centrifuge tube, and after this you get your plasmid band from where you can extract your DNA. So after, firstly what we did is isolation, then we did is ultracentrifugation. The next thing that we need to do is alkaline lysis. So it is another kind of a protocol that depends on the observation that at a specific alkaline pH the bacterial genomic DNA gets denatured, but your plasmids do not get denatured. So that is what we are trying to exploit over here using this alkaline lysis, is that Your bacterial genomic DNA gets denatured, but your plasmids do not. Now, what happens is that your subsequent neutralization precipitates the genomic DNA but leaves behind your plasmid, which stays in the solution. So, that is how you are actually extracting your plasmid. Now, the phages can also be used as vectors for cloning the DNA in the bacterial systems. Now, this phage DNA is then isolated from a pure suspension of phages recovered from the phage lysate. So, when we are extracting our plasmid, what we do is firstly we centrifuge it and get a pellet and all, and after that we dissolve in a solution, and then after that we do alkaline lysis where we are separating the so in the alkaline lysis, after we have done the alkaline lysis, we are separating the bacterial genomic DNA from the plasmids, and then after subsequent neutralization, the genomic DNA is precipitated, but your plasmid stays in the solution. And after this, what we do is we separate it out via your, firstly we wash it and then we separate it out from the using the different types of columns which we use, like silica binding column and all, and then we separate it with water. We elute it with water. Now comes to the cutting of the DNA. So the restriction enzyme CQR1 cuts a circular DNA molecule bearing one target sequence, resulting in the linear molecule with single stranded sticky ends. So we are giving examples of EQR1. There are other restriction enzymes as well which perform the same kind of function. So here we are seeing for EQR1 is that this is its recognition sequence, and then the cleavage occurs, and then we get the different sticky ends. Okay, but when there is a symmetric cleavage, say over here there is a symmetric kind of a cleavage, then we get is blunt ends. Okay, so we get blunt ends, so then we have to put a linker and all, and then we can join it, but the total functioning of this thing is absolutely same. Now coming to insertion, for that we have to choose a kind of a cloning vector in which we want to clone our gene of interest, and then a vector is any DNA molecule which is capable of multiplying inside the host to which our gene of interest is has been inserted for your cloning. Now in this process your restriction enzyme functions as scissors for cutting the DNA molecule. Now the ligase enzyme is your joining enzyme that joins the vector DNA with the gene of our interest, and this will produce our recombinant DNA. After you insert the DNA fragment into your vector and join it using the DNA ligase, then we can get our recombinant DNA. Now we have to introduce this vector DNA into the host cell. So we have the plasmid vector. Now the vector is added to a flask containing a culture of your E. coli. Now calcium ions usually form of in the form of calcium chloride is given to the flask, followed by a brief heat shock. So this is the entire protocol of your transformation, which I will also be discussing. Now what this helps, it allows holes to briefly appear in the cell surface of the membrane of the E. coli, making it permeable to the DNA and allowing the plasmids to enter. Okay, so after the different spaces have been created in the cell membrane of the bacteria, then your plasmid DNA can enter. So this is why the heat shock is very important at a particular temperature and for particular amount of time. If you're giving less amount of time, then it might happen that not all of the plasmids can enter your bacterial cell, and if you're giving for a more amount of time, then it might happen that your bacteria is only dying at such temperature. Then we have the other type of vectors is your phage vectors, which are introduced by the infection of bacterial lawn, which is growing on an agar plate. Now the culture or growth of viruses is made more difficult than the culture of bacteria or fungi by the fact that viruses will only grow. Now we have to place the gene in the vector. So we have our plasmid DNA. Now the DNA molecules are basically small, and these are separated based on their size. Now what we do is we break open these bacterial cells, and the chromosomal DNA is then centrifuged down, leaving your plasmid DNA in the liquid above the pellet. Now these plasmids are purified before cutting it with a restriction enzyme. So first you purify it, and then you undergo restriction digestion. We call this process as the restriction digestion, where you're cutting with particular restriction enzymes. Now these restriction fragments from your donor DNA are mixed with your plasmid DNA and then joined by their sticky ends. So the initial attraction is due to the hydrogen bonds, but the sugar phosphate backbone is then joined using an enzyme which we call as the DNA ligase. So in this manner we are placing the gene inside our vector. So this is the cloning into the plasmid. So we have a gene which is coding for your antibiotic resistance, and this is your plasmid where we have the restriction sites. Now these enzymes are used for cutting in the different specific positions, and then we have the foreign DNA or the region of our interest with the different cut sites, and then we get it. We have the sticky ends are being formed, and after this we undergo hybridization followed by your DNA ligation. Okay, so after the DNA insertion has occurred, we transform it into a bacterial cell. After this we clone it, getting multiple clones of such a particular bacterial cell containing our plasmids, and then these are plated on the medium, followed by first the antibiotic plate is being made, and then you plate the bacteria on it. Now only those bacteria which will contain our recombinant DNA will only growth, and after that we give a growth from, we pick up a colony and give the growth in your say LB culture or minimal media, in the different kinds of growth mediums and all, and after that we undergo DNA purification, that is your plasmid extraction. And after you get the plasmid, you again transform it in some kind of an expression vector from which you can get your protein of interest. Okay, let us take a few minutes of break, after which we can restart with transformation. Okay, so let us restart once again. Now we are basically left with only two main parts. One is your transformation, and another one is PCR, that is your polymerase chain reaction. And the last I will be discussing a few questions. So your bacterial transformation is in bacteria such as your E. coli, which can take up and express your foreign DNA, usually in the form of plasmids. So, in the gene cloning, using the bacterial transformation to make up lots of copies of your desired gene. So, this is your restriction enzyme which is cutting at different places, and then the join. The same thing is being repeated over here. Then you have the bacteria with the plasmid inside it, and then after you transform it and then you amplify it, you get the multiple copies of the identical genes present inside the bacteria. So the different steps of your bacterial transformation, the first thing is choosing a bacterial host. So it is very important to actually choose a very good model organism in which you are wanting to undergo, say, your bacterial transformation. So we basically use the E. coli as the model organism. So there are a few reasons of using E. coli. One being it is very well studied. There are no nuclear membranes present. It has the necessary uh, enzymes which are required for your replication. Then it grows rapidly, that is 20 minutes is its generation time. Then also it is inexpensive, normally not pathogenic, and it is easy to work with and undergo transformation. So these are the few factors which makes your E. coli um, and important, or the perfect candidate for your model organism, or the perfect candidate to undergo your bacterial transformation. The second step of bacterial transformation includes your choosing a plasmid which you want to transform. Now, there are different characteristics of a useful plasmid. One being it should have a single recognition site. So what does that mean? So plasmids only cut in one place so that it ensures that the plasmid is reformed in the correct order. So it is not like that you are cutting your plasmid in the different places and you chop it off. It is like cutting your plasmids in the specific position such that you get this plasmid, or you can insert your gene of interest in that particular position only, and not like that you are inserting a gene of interest in any position. Okay, so it is necessary to have a single recognition site. Also, it is necessary to have your origin of replication. So what does this origin of replication do? It allows your plasmids to replicate. So it's the origin of replication. So where your replication actually originates from or starts from. So it allows the plasmid to replicate and make copies for the new cells. Okay, so this is the other thing which important thing. The next thing being your marker genes, that is, it identifies the cells that have been transformed, like the gene for your antibiotic resistance, that is, your bacteria is plated on a media with an antibiotic, and only bacteria that have taken up a plasmid will only grow. And the other thing is that the gene expresses color. Now the bacteria that have taken up a recombinant plasmid are a different color than bacteria that have not taken up non-recombinant vector. So now if you are actually incubating your plasmid or inserting a fluorescent type of a thing inside your plasmid, then you can see the colonies which are colored, and only those bacterial cells which are colored you can pick up from that. Okay, so that is another thing. And the other thing being that only the genes which grow in the antibiotic resistance is your plasmid of interest, contains your gene of interest. So these are the three things which are very important, and these needs to be kept in mind, especially for your exams, and these are the basic requirements of our plasmid. First being your single, first being your single recognition site. Why do you require this? Because it only cuts in one place. The second being your origin of replication, that allows your plasmids to replicate and make copies for the new cells. The third one being your marker genes, that identifies your cells that have been transformed. So one point being via your antibiotic resistance, the other being that your gene expresses color. Now the third step is that the bacterial, preparing your bacterial cells for the transformation. So you have to make different cells, or the cells. You have to make the cells competent such that they are able to withstand the different changes in the heat, temperature, and are also being able to take up your plasmid. So what do we do to prepare ourselves? So it is like a war that you are taking up plasmid. So you have to prepare yourselves for such difficult situations. Okay, so what do? You treat with calcium chloride. So first being you treating with calcium chloride. So what it does, it requires this calcium, this divalent ion, so it softens your phospholipid bilayer. So it is required for softening your phospholipid bilayer of the cell membrane, which allows your plasmid to pass through. So if you are softening it, and then when you're giving it some kind of a heat shock, the cell membrane creates some kinds of holes or spaces for your plasmid to enter. The other process being your electroporation, that is you're giving a very brief electric pulse. And the third one is that you directly inject the plasmid into the bacterial host. So we do either A or B. We mostly do not directly inject it. Now depends on the different conditions of the different host organisms and all. So these are the three ways. Now the fourth one is your plate transformation on appropriate media, which contains your nutrients for the bacteria and also the antibiotic that will help you to distinguish between your transformed bacteria from the non-transferred bacteria. And then what you do is incubate the plates overnight. And we know that E. coli grows well in the body at 37 degrees centigrade. So we incubate in 37 degrees centigrade, say a plate incubator, and then we analyze the plates after we have got the colonies. So this is your entire transformation chart. So in the first step we have said that isolating of your plasmid or your vector DNA and the human DNA. Okay, now this plasmid has the different ampicillin resistance, origin of replication, the different multiple cloning sites, and your lag Z gene. After that you cut the DNA with the same restriction enzyme, and it has forms the sticky ends. In the meantime, your cell contains your gene of interest. You have the DNA containing the gene of interest. You cut them with the same restriction enzymes. Now what you do is you insert the human DNA into the plasmid and you mix the DNAs. They will join by base pairing. After you have done this, you add the DNA ligase to join them covalently. So DNA ligase is what it does, it joins them covalently. Now what you see is, now you introduce the plasmids into your bacteria via transformation, and then you plate them on the then you plate them on the agar plates, and after that you give culture. From there you isolate your plasmids, and then you can either undergo your expression test inside your BL21 or the other expression vectors, or you can send this plasmid for sequencing to check whether they have inserted well or not, and whether your cloning has been successful or not. Now coming to our last topic, which is our PCR. So it is known as the polymerase chain reaction, which was first described in 1971 by Clape and Corona, and then redescribed and successfully used in 1985. So this allows your massive amplification of specific sequences that have defined endpoints, and this is a fast, powerful, adaptable and simple, sometimes simple technique. And it has various wide the number of applications in this biological field. So why do we need to amplify the specific sequences? So the first one being we need to obtain materials for cloning and sequencing, or if we need to undergo in vitro studies. So for that we need to amplify the specific sequences. Also we need to amplify these specific sequences if we have to say monitor the gene expression, or if we want to diagnose a genetic disease, like what are the mutations or the recombinations which have occurred. If so, then what is the disease, which is under what kind of mutations are such a disease occurring. So it is also important, for that we need to amplify the specific sequences. Also, if we have to verify the identity of the DNA constructs which we have engineered in our laboratories, and we are doing it, so for that we need it. Also it helps to identify an individual and reveal the presence of any kind of microorganisms. And also these are the different things. So first one is obtaining your materials for cloning and sequencing or for in vitro studies. Next one is verifying the identity of the engineered DNA construct. The next one is monitoring gene expression, diagnosing a disease, revealing the presence of a microorganism, identifying an individual. So these are the different reasons why we need to amplify the your specific sequences. Now what we need for PCR is we need the template DNA, that is your plasmid that contains the target sequence or the region which we need to actually amplify and all. Also we need the primers which we design on our own. So these are short oligonucleotides that define the ends of the target sequence, and these are the portions, these are the primers which have a particular portion of your target sequence, and these are responsible for actually amplifying your entire template sequence or the entire gene of your interest or the entire DNA sequence which you're going to insert into the vector. Then we require an enzyme which is your thermostable DNA polymerase, can be your Taq polymerase, which has the ability to withstand higher temperature, or you can use the other enzymes like your fusion. So all of these have can have an optimum works at an optimum temperature of your 72 degrees centigrade. So these are basically important. And after that we have the different kinds of buffers, say your GC buffers or your HT buffers, and we need the DNTPs, which are very important for the addition of the nucleotides in the particular target sequence in the time when we are amplifying. And finally we need a thermal cycler where we have to place our mixed solution, and then the thermal cycler will undergo different temperature changes, giving us your denaturation step, annealing and optimize elongation steps and all. So how this PCR works? So it is a simple PCR. So it is a simple PCR reaction in the first cycle. So what happens is that you have a double-stranded and DNA target, which can also be a single-stranded, so that does not matter much. So after that you place it in the thermal cycler after putting all these necessary ingredients or the component inside that mixture, and after that you denature it at higher temperature, can be 94, 95, 98 depending on the type of enzyme or the type of reaction you want to set up. So this happens at a higher temperature, and then you undergo primer annealing. Okay, then annealing happens where you have the two primers, and this moves in this direction, the reverse primer moves in this direction, and it amplifies. Okay, so the amplification process occurs at a temperature. So when you are preparing or designing your primers, you have to check the TMs. So these are the melting temperatures. So the temperature at which your DNA is half denatured and half natured is known as your melting temperature. So this Tm actually varies. Okay, so this variation can be so you set the annealing temperature at Tm minus 5 degrees centigrade. Okay, so this is like that, can be 4, can be 3. Now you need to optimize that depending on the products which you are getting, the non-specific bands if you are seeing any. So depending on that you need to do that optimization thing, but as of now you can say that as Tm minus 5 degrees centigrade. And then after that you have to undergo your DNA synthesis, that is your primer extension. So this is the extension phase, and after that the same thing occurs. So in after these 3 steps, so you see the temperature of this primer extension is 72 degrees centigrade, where your enzymes actually starts working and they undergo the primer extension and all. So there are 3 steps. First is denaturation, the next one is primer annealing, and then your primer extension by the DNA polymerase. And then the second reaction occurs in the same manner. So denaturation followed by extension, annealing followed by extension, and then the third step continues. So at the end of say 29 or 30 cycles, you would be having around a big number of your plasmids or your template sequence. So this will actually help you to have multiple copies of your template sequence, which you can actually utilize for the different future studies. Okay. So that is all about the polymerase chain reaction, the transformation, and the recombinant DNA technology. Now I have a few questions. So just because we do not have, I guess we do not have any students present in this lecture, so I would just only discussing with you the questions, some practice problems which will help you in solving your assignments and the exam which is coming up. Okay. Okay, so the first question, it says that your plasmids can be successfully used as vector for gene cloning as. So what are the options given? One is that they are extrachromosomal, small circular double-stranded DNA. They have origin of replication. They possess selectable marker. So if you go through my lectures, you can see that um, the plasmids which can be successfully used as vectors needs to have 3 things. One is that your origin of replication is important. They needs to have a selectable marker, which is your antibiotic resistance it needs to have, and they have to be extrachromosomal, small circular double-stranded DNA. So all of the above statements are correct. Okay. The next question says, to cut a DNA molecule, which of the following enzyme is used? The ligase enzymes, the phosphatase enzymes, the polymerase enzymes, or the restriction enzymes? So I was teaching for so long. These are the restriction enzymes which cuts at the specific sequences, either giving rise to your sticky ends or the blunt ends, after which you cut your DNA fragment with the same restriction enzymes and then insert it into the vector, and after that you get the plasmid and then you transfer it. Okay, so we use the restriction enzymes to cut the DNA molecule. We use ligase enzymes for joining. Okay, the third question says the DNA molecule to which gene of interest is inserted for transferring it to other organism is called. So what do we call it? This is a very easy question. I don't need to discuss it much. We do not call it transporter, exporter, or converter. Hello, why are you so late? Class is about to end. It has started from 6 o'clock. Were you not aware of the timings? In anyways, I was just discussing with the questions and all. So after that, I would be ending my lecture. So in anyways, you. So would you like to answer a few of the questions? Possibly I do it in my every class, just because today I did not have any students, so I started with the question and answers. So you can try if you want to. In any ways, I would be discussing only. So, so I was with the first question. So the first question was asking plasmids can be successfully used as a vector for gene cloning. So it says that the plasmids which are extra chromosomal, circular, double-stranded, having origin of replication and possessing different antibiotic resistant and selectable markers are the ideal candidates for your which can be used as vector for cutting the DNA molecule. The enzymes which we use are restriction enzymes. Okay, so you can try with the third one. So these are the two questions I just started discussing. The DNA molecule to which the gene of interest is inserted for transferring it to another organism is called. What do you think is the answer? You can write it down in the chat box. Yes, that is correct. So 3B is the right answer. We call that as the vector. Okay. The next question says to join the gene of interest with the vector, which of the following enzymes is used? Now for joining the gene of interest to the vector, which of the following enzymes do we use such that it joins? So yes, we have ligase, lyase, ribonuclease and polymerase. Yes, we use DNA ligase for the joining purpose. Yes, correct. Now question 5 says the mechanism of intake of DNA from surrounding environment by the cell is called. So you're taking DNA from the surrounding environment. What do we call it? Can you tell me what is transduction or conjunction? Do you know what are these terms? In any ways, option C is correct. That is, when you are taking DNA from the surrounding environment, it is called the transformation. But your transduction is it is the process by which a virus transfers the genetic material from one bacterium to another. Okay. And when you're taking up the DNA, when the DNA is being transferred between the bacteria through, you call a tube like structure, which we basically call as the sex pillars or a pillars or a structure in which the DNA is transferred between the bacteria through a tube. We call that as conjunction. But when we take the DNA from the surrounding environment, it is called a transformation. So 5C is correct. What is number 6? What is the role of selectable marker in a cloning vector? This is an MSQ question. So what is the role of a selectable marker in cloning vector? Yes, that is correct. That it helps in the maintaining of the vectors inside the cell, and also it helps in the selective survival of the cells that have the vectors, and only those bacterial cells are going to survive that have taken up your recombinant yeah, plasmid and all, but others will either die. And if you are not using say a selectable marker, but in the other way, if you are actually say inserting some kind of fluorescence inside your vector and all, or tagging it with some kind of fluorescence, then your bacterial cells will, when they will grow, they will actually fluoresce or show some kind of color and all. Okay, but in any way 6A and B is correct. Okay, the next question says multiple cloning site in a vector has. So what does your multiple cloning sites in a vector has? Yes, it contains unique restriction sites. Correct. What will happen if the origin of replication is missing in a vector? What will happen if your site is not present? Yes, the vector, just because the replication site is not present, so the vector will not replicate. So 8D is correct. And the 7, yes, 7C, 8D, correct. Okay, question number 9. A large number of copies of a gene can be obtained following. How can you make multiple copies of the gene? Yes, we need to undergo polymerase chain reaction. So that helps in the generation of multiple copies of the gene. And I was showing in the previous slides, after 29 to 30 cycles, you get numbers in 10 to the power 7, 10 to the power 8. Okay, so that is correct. 9B. Question number 10. Which of the following statement is true? PCR machines are known as thermocyclers. Exponential amplification of a gene can be done from very small amount of DNA. Thermostable DNA polymerase is absolutely necessary, or all of the above. Yes, all of the above are correct, because as I was showing, for PCR you require a thermostable DNA polymerase, you require the primers, you require a thermocycler, which is a PCR machine for modulating the temperature change, and also exponential amplification of a gene can be done from very small amount of DNA. Like from 1 we get 2, then 4, 8, so it's an exponential way it's increasing. So the first step in PCR is known as the, these are easy questions. So what happens? Yes, the denaturation. The next step being your annealing, and the last step is your extension. To which side of the primers the restriction sites are included for cloning? 5' end, 3' end, both the ends are not present in the primers. To which side of the primers are your restriction enzymes included? No. So see, so they are included in the 5' ends only. So because if you see even in the complementary sequence, so it seems like that they are present in the 3' end, but from the complementary sequence, if you see, they are present in the 5' end. Why it is so? Because these restriction sites are actually, or these enzymes identify these restriction sites from the 5' end to the 3' end, And so when you are actually designing your primer, you place them in the 5' end such that the enzymes can read them in the 5' end to the 3' end pattern. And when you're designing a complementary primer, that is a reverse primer and that, so you also do that in the 5' end to the 3' end, and your bacteria is going to read in the same pattern. So that's why we always place the restriction sites in the 5' end. So 12, A is the right option. Okay. So the last, I think that's the last. Yeah. So the stop codon is present in the, so where is your stop codon present? Okay, so if it is present in the 3' end of the forward primer, will your primer be able to anneal or extend? Will it be able to perform any reaction as such? No. But then where should it be present? It should be present in the 5' end of your reverse primer. So because it is your complementary, so it is at the back end of your back end side. So when it will be present in the 5' end of your reverse primer, then after the entire process of your annealing and your elongation, only then it will reach the stop codon, and then the thing stops. Even during your what you call is protein expression, even in that time, then now you have the entire gene sequence, and you know that you have the entire DNA sequence, and you know what type of protein you're going to get. So when if it is present in the 5' end of the reverse primer, so after it reads from the 5' end to the 3' end in the forward direction, it reaches the stop codon after the entire protein has been expressed. So that's why your stop codon is present in the 5' end of the reverse primer. Okay. The next question is, so 13, C is the right answer. Now question number 14. In PCR, the DNA synthesis takes place at. So what temperature does the DNA synthesis takes place? Yes, 72 degrees centigrade. That is correct. Now the last question. Expression vectors differs from cloning vectors by having. What does these expression vectors have or do not have? Or how are they different from your cloning vectors? Yes. So the name says it all. The expression vectors differs from the cloning vectors by having the different gene expression control elements, which will be playing an important role during the expression of your gene and getting your gene product, that is your protein. So yes, 15B is the right answer. Okay, so that's all from my side with the questions. Okay, thank you. If you people do not have any questions.